The digestive system's function is to break down ingested food into molecules small enough to be absorbed and utilized by the body. In previous lessons, we explored the control of the gastrointestinal GI system, the motility of its regions, and the secretions of its glands. In this lesson, we'll focus on the digestion and absorption of proteins, lipids, and carbohydrates. Digestion and absorption are distinct processes, but they are closely related. Digestion is the process by which macromolecules are broken down into micromolecules, whereas absorption is the passage of digested molecules across the intestinal epithelium. If food is not digested, it cannot be absorbed, and it is of no use for digestion to occur if the nutrients cannot be absorbed. In this lesson, we will focus on non-fermentative processes, that is, those that occur in non-ruminant animals, such as dogs, cats, and others. In other lessons, we will explore fermentative processes in greater detail. Here, we'll study chemical digestion carried out by the animal's enzymes, although digestion also includes the physical breakdown of food. Physical breakdown facilitates the movement of food through the digestive tract and increases exposure to digestive enzymes. This begins in the mouth with chewing and continues in the distal stomach, where strong contractions further break down macromolecules. In the stomach, food is exposed to hydrochloric acid, and protein digestion begins with pepsin, secreted by chief cells. Only particles smaller than 2 millimeters pass into the duodenum, meaning the stomach contents are ground into a near-liquid state. The final phase of digestion and absorption occurs in the intestine, primarily in the duodenum. To understand this, we need to examine the intestinal structure that enables these processes. The small intestine's mucosa is specially designed with 3 levels of projections to increase surface area for contact with food. The first level consists of circular folds, which are not present in all animals. The second level involves villi, finger-like projections of the mucosa found across all species. At the base of these villi are crypts, and both are covered by epithelial cells called enterocytes. The third level comprises microvilli, cytoplasmic projections of enterocytes, forming a brush border. These 3 levels exponentially increase the mucosa's contact surface with the food. Enterocytes have an apical membrane facing the intestinal lumen, where microvilli are located. These are associated with glycoproteins, including enzymes for digestion and transport proteins for absorption, collectively forming the glycocalyx. The basolateral membrane, on the other hand, encompasses the cell's sides and base, facing away from the lumen. Tight junctions connect neighboring enterocytes, allowing only water and electrolytes to pass between them while other molecules are blocked. The space between enterocytes is known as the lateral space, and adjacent to these cells are blood and lymphatic vessels. Nutrients are absorbed across the apical membrane, pass through the basolateral membrane and lateral space, and then enter these vessels. Depending on the nutrient type, it may enter either the bloodstream or lymphatic system. Goblet cells, interspersed among enterocytes, secrete mucus that covers the mucosal surface. This mucus combines with the glycocalyx to form a viscous layer that traps molecules approaching the surface. As we discussed in the previous lesson, digestive glands secrete numerous substances, creating a liquid environment. Thus, digestion and absorption occur in an aqueous medium. Water near the mucosal surface flows more slowly due to friction with the viscous layer, forming a stable water layer. This is similar to how water in a stream flows faster in the center and slower along the edges. This stable water layer, together with the viscous layer, creates a diffusion barrier for nutrients passing through the surface. With this structural overview of the small intestine, let's delve into the processes of digestion and absorption. Chemical digestion by enzymes involves hydrolysis, the breaking of chemical bonds by adding water. Digestive enzymes fall into 2 main categories: those that act in the intestinal lumen and those attached to the enterocyte membrane. Luminal enzymes are secreted by glands such as the salivary glands, gastric glands, and primarily the pancreas, mixing with the food content. Enzymes attached to the enterocyte membrane form part of the glycocalyx, completing molecular breakdown. Digestion thus has 2 phases: the luminal phase, performed by luminal enzymes, and the membrane phase, performed by enzymes attached to enterocytes. This membrane phase digestion occurs within the microenvironment formed by the stable water layer, mucus, and glycocalyx. This lesson will focus on the digestion of the main macronutrients: proteins, lipids, and carbohydrates. Carbohydrates, in turn, are divided into sugars, starches, and fibers, though fibers will be addressed in a separate lesson. We will begin with the digestion and absorption of carbohydrates. Carbohydrates are composed of repeated simple sugar molecules. Simple sugars are monosaccharides. From these, we have disaccharides, trisaccharides, oligosaccharides, and so on. Therefore, there are several types of carbohydrates, and among all of them, starch is the only one that needs to undergo the luminal phase, while the others are digested in the membrane phase. The enzyme responsible for starch digestion is amylase, which is secreted by the pancreas and by the saliva in some species. Amylase breaks starch down into polysaccharides, trisaccharides, and disaccharides. These products then move to the membrane phase, along with the other carbohydrates. In the membrane phase, there is a specific enzyme for each polysaccharide. Thus, we have maltase, sucrase, lactase, and several others. All of them are converted into monosaccharides before absorption. I will not go into detail about the types of transport involved in the absorption process. To simplify, the molecules pass through the apical membrane, the basolateral membrane, the lateral space, and then enter the vessels. Proteins, composed of amino acid chains, are digested by several luminal enzymes, mainly pancreatic ones, but also those secreted by the stomach. As mentioned earlier, these enzymes are produced and secreted in inactive forms, or zymogens, to prevent them from digesting their own cells. In the stomach, pepsinogen is activated by hydrochloric acid to form pepsin, which is adapted to the stomach's low pH. In the duodenal mucosa, enteropeptidase activates pancreatic trypsinogen into trypsin, which initiates a cascade activating other protein-digesting enzymes. Proteolytic enzymes are classified as endopeptidases or exopeptidases. Endopeptidases, such as pepsin and trypsin, break internal peptide bonds, creating shorter chains, while exopeptidases cleave terminal bonds to release individual amino acids. After luminal digestion, peptides move to the membrane phase, where further breakdown occurs, yielding dipeptides, tripeptides, and free amino acids. Unlike carbohydrates, proteins can enter enterocytes as dipeptides or tripeptides, where intracellular enzymes convert them into free amino acids. Only free amino acids cross the basolateral membrane into the bloodstream. Now for lipids, the main dietary lipid is triglycerides, though others like cholesterol are also present. Unlike other nutrients, lipids are hydrophobic and insoluble in water, posing a challenge in the aqueous digestive environment. Lipid digestion involves 4 steps: emulsification, hydrolysis, micelle formation, and absorption. Emulsification begins in the stomach, where lipids are warmed and agitated in the distal stomach, breaking down fat globules before reaching the duodenum. In the duodenum, bile acids, a key bile component, complete the emulsification process. Hydrolysis is then carried out by pancreatic enzymes, lipase and cholipase. Lipase, secreted in its active form, cannot access emulsified lipid droplets due to the bile coating. Cholipase helps by clearing a path, enabling lipase to break triglycerides into fatty acids. These fatty acids combine with bile acids to form micelles, which are water-soluble structures that diffuse through the stable water layer to the enterocyte membrane. Micelles deliver lipids to the apical membrane of enterocytes, where absorption occurs. Bile acids remain in the lumen and are later absorbed in the ileum, re-entering the liver via the portal circulation in a process called enterohepatic recycling. Once inside enterocytes, lipids are packaged with proteins to form chylomicrons, spherical structures with a hydrophobic core surrounded by hydrophilic proteins. Chylomicrons cross the basolateral membrane into the lymphatic system rather than the bloodstream, completing lipid absorption. As mentioned earlier, after lipid absorption, bile acids remain in the intestinal lumen. When they reach the ileum, there is a specific mechanism that allows these molecules to be reabsorbed. Once absorbed, they enter the portal circulation and are transported back to the liver. When they arrive at the hepatic sinusoids, they are taken up by hepatocytes and reused for the production of new bile. The liver is extremely efficient at removing bile acids from portal blood, so only about 5% actually reach the systemic circulation. This recycling process happens several times throughout the day. The continuous movement of bile acids between the intestine and the liver is what we call enterohepatic circulation. And that's where we'll stop for today. Before you go, don't forget to like the video and subscribe to the channel so you don't miss any of the upcoming classes. See you in the next class.